All right, hi everyone. This is the video lecture for the topic protein function and regulation. To start us off, I just want to give us a quick reminder of where we left off in the last topic. Recall the last topic was talking about the four classes of organic molecules, and the fourth and final class that we talked about were the proteins. Recall that we said the proteins are the most diverse and complex group of macromolecules in cells in terms of their composition and structure, which we talked about. And we said that this is also true for protein function, but we said we were going to leave our discussion of protein function until the next topic, and that's where we are right now. Another thing I want to mention is I'm going to break this topic up into 5 short video lectures in case it's convenient for you to focus on one or a couple particular ones. In this first one, I'm going to talk generally about the types of functions that proteins can carry out in cells. After this, we're going to talk about 4 different types of regulation that cells use to regulate proteins, and I'm going to break each of those 4 types of regulation up into an individual video lecture, a short one. Okay, so to get us started over here, what I'm going to do is I'm going to build a list of different types of protein function and talk about each one as we go through. The first general way in which some proteins function in cells is as biological catalysts, in other words, as enzymes. And you've talked about this before, if at least in Biology 121. Recall that enzymes refer to biological catalysts that increase the rate of chemical reactions by reducing the activation energy that is required for the reactions to proceed. Shown down here is a graph that illustrates this point, and it's the graph that is typically shown to make these points. Recall the way you read this is on the x-axis. It is showing you the progress of some chemical reaction in which a substrate, in this case just some generic substrate, is being converted into a product. On the y-axis, it is trying to show you the energy state of the molecule as it begins from a substrate and proceeds through being a product. If you look at the energy state of the molecule as it goes from substrate to product, you hopefully recall that what happens is the substrate has to increase in its energy state, and that energy state will increase until some peak that is referred to as the transition state, after which the energy state drops all the way down until it reaches the final energy state of the product. Recall that the amount of energy that has to go into the reaction to get the substrate to its transition state can be quantified, and refer that we call that amount of energy the activation energy of a chemical reaction. Okay, so with that quick reminder, this graph here, this line, is intended to represent the activation energy required for some hypothetical uncatalyzed reaction, and to try and bring enzymes into this. Right? What this blue dotted line is trying to show you is generally how enzymes work. Right? What enzymes do is if they're going to catalyze a reaction, they will take the substrate and basically get it to its transition state, which requires a lower activation energy. And so in turn, that accelerates the rate at which the substrate will get to its transition state and then proceed to its product. And so this is fundamentally how enzymes accelerate chemical reactions. I also want to remind you that enzyme catalysis takes place through 3 fundamental steps. In the first step, what has to happen is the enzyme needs to bind to its substrates, right? So we might say enzyme substrate binding has to happen first. Remember that this takes place at a specific site within the enzyme's or the protein's tertiary structure, which is called the active site. So the active site is the location in the tertiary structure of an enzyme that binds to their substrates and catalyzes chemical reactions. And so the enzymes and substrates will bind, resulting in what's called an enzyme-substrate complex, as is shown here. Once they're in complex, the enzyme is going to facilitate the progression of the substrates to their transition state and actually catalyze the reaction. And so we might summarize that by saying the second step is reaction catalysis. And then once the reaction has been catalyzed, the product gets released. Right? And so as you can see in this example here, this generic example, in this reaction, this enzyme is taking 2 separate molecules, 2 separate substrates, and catalyzing a reaction in which they are getting linked up into one molecule or one product. Okay, so that's sort of the 3 steps to just keep in mind. And one of the key things to remember is that when an enzyme catalyzes a reaction, it has to physically interact with its substrates and make that reaction go. Another thing I want to remind you is based off of this timeout up here, if you will, and it's this question of what is this brown blob here, right? And the point that I just want to make sure that you are trying to stay conscious of is, again, we're going to look at a lot of images of proteins that look like blobs, but try to keep track of the fact that when you see these blobs, what they're really representing are polypeptides folded into a particular tertiary structure, right? And so we might, instead of using a blob, use something like this, right? Which would be a more accurate representation of a protein. But as we move through the course, a lot of times we are going to depict proteins like blobs, even though you want to try and keep track of what they really represent. Okay, so with that in mind, I want to make 3 points here, or ask 3 questions here to get you thinking about that is going to hopefully help you understand cell biology as we move forward. One question I want to ask is, are enzymes specific or non-specific for their substrates? And I'm sure you know that they are specific, right? So each particular enzyme is going to catalyze reactions on specific substrates. The other thing I want to ask you is, what dictates their specificity? In other words, what dictates the specificity of a particular enzyme for its substrates? And really we can say that there's 2 general things that dictate the specificity. On the one hand, we can say it's the active site conformation, which of course refers to the shape of the active site and how that shape fits the substrates. But there's another important part to this this answer that maybe you haven't thought about, and that is that another key part is the amino acids and their properties that are lining the active site of an enzyme, right? So just take a second here and look at what I'm going to try and show you. Imagine in this hypothetical example, at this particular site in the enzyme, imagine lining the active site there is a basic or positively charged amino acid, and imagine that the site where the substrate comes into the active site right there, imagine there's a negative charge. Hopefully you can appreciate that the positive and negative attraction could have an important effect on the ability for this particular substrate to bind to the active site. Likewise, imagine down here at this area of the active site, imagine that there is a nonpolar or hydrophobic amino acid that is toward the active site, and imagine on this substrate there is a hydrophobic nonpolar area. So hopefully you can appreciate that if that's the case, perhaps hydrophobic interactions between these 2 areas could also facilitate the binding of this substrate into the active site. Okay, so very oftentimes amino acids and their properties and how they complement the chemical properties of the substrates are also really important. A third question I want to ask you is, how do you think mutations would affect the activity of this enzyme? In other words, how do you think mutations within changes in amino acids within the enzyme might affect this reaction? So let's say, for example, remember this positively charged amino acid, this hypothetical positively charged amino acid as shown here. Imagine that this enzyme was mutated so that instead of there being a positively charged amino acid, there was a negatively charged. So the question I want you to think about is, do you think that that change to a negatively charged amino acid would have any impact on the ability of this enzyme to bind the substrate and or catalyze the reaction? Or alternatively, think about this amino acid down here that we said, imagine is hydrophobic. Imagine that that amino acid got mutated or changed into a polar amino acid, right? Do you think that if that amino acid was now polar instead of hydrophobic, do you think that would have any effect on the ability of the enzyme to bind to the substrate and or catalyze the reaction? And third and lastly, remember we said that hydrophobic amino acids not always, but most often, are sort of in the center, buried in the core of enzymes of protein structure. So imagine that there's a hydrophobic amino acid in the core of this enzyme, and imagine that that amino acid got mutated or changed into a charged amino acid. Do you think that would have any effect on the enzyme, its ability to bind its substrates and or its ability to catalyze a reaction? So we'll talk about this in class, but if you can, start trying to think about these types of concepts. And of course, the answer to this question here is absolutely mutations could affect the enzyme's ability to interact with the substrates and carry out reactions. Okay, so that's the basic overview of biological catalysts or enzymes that you want to start trying to sort of refresh your memory of. And a key thing to appreciate here is a reason why understanding enzymes is so important is because enzymes catalyze nearly every chemical reaction that takes place in cells. And if I were to say, can you think of any examples? One set of chemical reactions that I know you've thought about pretty extensively in general biology are the chemical reactions of metabolism, right? Of glucose catabolism. So recall glycolysis and the citric acid cycle. Remember, those are the metabolic pathways through which cells take glucose, right, and catabolize it as part of the process to generate ATP. Now, the point here is not to memorize these chemical reactions at all, but the point is to look at them, try to generally remember them, try to remember that all of these chemical reactions are dependent on specific enzymes, right? And now I've shown those here in red, right? For glycolysis, all the enzymes that catalyze each reaction are shown in red. And each of the chemical reactions of the citric acid cycle are also dependent on enzymes, even though they're not shown in this image, right? And so I just put this up here to try and remind you of these concepts and to get you to again, think about and appreciate the critical need for enzymes in cells. Okay, so that is a reminder of the fact that many proteins function as enzymes. But another function that many other proteins carry out in cells is to function as what we might call structural proteins. What I mean by this is take a look at these three different types of cells that we all have many million of in our bodies, right? Cardiomyocytes, which are heart cells, fibroblasts, which are present in a bunch of tissues, and neurons that are present in our brains. What I want you to appreciate is the fact that while these are all cells and they have a lot in common, they have vastly different shapes or structures. And so the question is, how do these different cells, even though they're composed of the same basic stuff, how do they take on such intricate and different structures? And the answer is that there are structural proteins that function to give cells their structures. For example, we're going to talk about proteins called microtubules, which are one class of structural proteins, which are shown here in yellow, and they play critical roles in giving cells their specific shape or their specific structure. Okay, so some proteins function as structural proteins. A third function that yet other proteins have is to function as what are called scaffolding proteins. Now, I suspect you haven't heard of the term scaffolding protein before, so I'm going to give you a sort of quick definition. So we can say that scaffolding proteins refer to proteins that function to recruit or coordinate specific proteins to a specific place in the cell, right? And so what we mean by recruit is basically these scaffolding proteins are going to physically bind to some other protein and help it get to a location in the cell. One example I like to bring up, because you've talked about it in general biology, is the protein RNA polymerase. And this is the image here that you looked at in general biology, if you took it at least at Bridgewater, showing you this RNA polymerase here as the sort of gray guitar pick, if you will, that is carrying out transcription. The key thing to appreciate is that it turns out that RNA polymerase, when it carries out transcription, is absolutely dependent on a very large group of scaffolding proteins that bind to RNA polymerase and actually escort it to the locations where genes are that need to be transcribed. Right? So RNA polymerase needs help from scaffolding proteins to quote-unquote find genes. In other words, find DNA sequences that it needs to transcribe. And so rather than looking at a more simple image like this, this is a more accurate depiction of RNA polymerase, shown here in purple, that is loaded onto double-stranded DNA here in brown, ready to transcribe a gene. And all of these other variously colored blobs represent what we might call scaffolding proteins that bind to RNA polymerase and take it to DNA sequences where genes are located so that it can carry out transcription. So we're going to talk about how this works, but for now, the point is to just appreciate that there are many scaffolding proteins that carry out this fundamental role of getting proteins to the correct spot. Okay, so scaffolding proteins is another thing. A fourth function that many other proteins carry out is to serve as what we can call transmembrane channels or transporters. To think about this, I just want to give you a reminder of something that you've thought about many times now, right? And that reminder is that the phospholipid bilayers that make up cell membranes, recall, are semi-permeable. which means that there are some molecules capable of passing directly through them, whereas there are many other molecules, large polar molecules and ions, that are absolutely incapable of passing through the phospholipid bilayer. But nevertheless, the cell is critically dependent on getting many of these molecules from outside to inside of them. And so the question is, how do they then manage to get these molecules across their membranes? And the answer to this very often is that cells have membrane-embedded proteins, or transmembrane proteins, that function to transport molecules across. And so shown here is an example, just a schematic depiction of a transmembrane channel protein that transits through the plasma membrane or through a phospholipid bilayer and has a channel in it that allows a passageway for molecules that might otherwise not be capable of getting across the membrane. Shown here is a more accurate depiction, a ribbon diagram of such a channel protein. The name of this is aquaporin. Nothing to memorize, it's just to show that it's a specific example of a specific structure of a channel protein that transits a phospholipid bilayer, and this molecule here is showing a molecule that is passing through it and across the membrane. Okay, so there's transmembrane channels and transporters. And then the last specific example I'm going to tell you about is that many other proteins function as what we call transmembrane receptors. And we're going to talk a lot about transmembrane receptors in the final part of the course. For now, let me just give you a general sense of what they are. So here's just a quick definition. We could say that transmembrane proteins, they are proteins that, on the one hand, are going to detect the presence of some extracellular molecule, right? So extracellular molecule means it's going to detect the presence of some molecule that is outside of the cell. And in response to that, they stimulate chemical reactions inside of the cell that in turn induce a behavioral response. And so for an example of what I mean by this, I'm going to use one example, which are transmembrane receptors that we call growth factor receptors, and how growth factor receptors cause the behavioral response of cell proliferation. So imagine here, to try and explain what I mean here, imagine this is some cell, right? And hopefully you know that if I were to say, imagine this cell undergoing proliferation, hopefully you remember, well, what that means is that this cell is going to divide into two cells, right? That is what we mean when we say cell proliferation. But the other thing to appreciate is that it turns out whether cells proliferate or not is controlled largely by a group of molecules, extracellular molecules, called growth factors. And these growth factors are secreted typically by neighboring cells to regulate the proliferation of other cells. And so imagine we have a situation like this, where within some tissue in your body, there are two cells, one of which has produced a growth factor molecule. And what this cell will do is it will secrete or release that growth factor. Well, if that growth factor makes it to the general vicinity or the environment of the other cell, and if that cell has one of these transmembrane receptors, which we might call a growth factor receptor, that specifically binds to that growth factor on the extracellular side of the protein, well, it turns out what these receptors do is once they bind to the growth factor within the cell, they stimulate a series of chemical reactions, which is what I'm trying to show with these sort of yellow things, right? The key is it's the stimulation of these chemical reactions in response to the growth factor binding the receptor that in turn is actually what drives the cell to proliferate. So at the end of the course, we're going to talk in pretty good detail about exactly how this works out. But for now, I just want to give you a sense to appreciate this idea that some proteins function as these receptors that are binding some molecule, some extracellular molecule, and once they're bound, they activate chemical reactions that can in turn induce a behavioral response. In this example, the behavioral response was the induction of proliferation. All right, so then we have transmembrane receptors, and then we could, to be honest with you, we could probably spend an hour going through other specific examples of functions that proteins play. But I think this should suffice to hopefully start giving you a sense of the types of things that proteins do, and a lot of them which we're going to talk about as we move on through the course. All right, and a final essential property that I want you to appreciate about proteins, other than the fact that they are super complex in structure, composition, and function, a final essential thing to really appreciate about proteins is that they are regulatable, right? What I mean by this is enzymes. Because they're proteins, they can be turned on and turned off, which in turn can regulate whether or not glycolysis and the citric acid cycle, for example, are taking place. Structural proteins that give cells their structures, they are capable of being remodeled, which in turn can drive shape changes in cells. Protein channels and transporters, as this image now is trying to show, these as proteins are capable of being either closed so that things cannot pass through them or opened to allow things pass through them, right? And so this type of idea in terms of thinking about all of those protein functions that we talked about, they can all be modulated or regulated in cells, right? And it's absolutely critical that that is the case so that cells work the way they should. And so with that in mind, our next overriding question is going to be, what are specific mechanisms that cells have in place to actually regulate proteins? And that brings us then to sort of the next area, right? Now we're going to talk about protein regulation. And what we're going to do is we're going to talk about 4 different mechanisms that cells use over and over to regulate protein activity, and 4 different mechanisms that we're going to talk about over and over again in this course, which are listed here. And so the next video lecture will be a short lecture on this mechanism here, allosteric regulation, and then the others will go through the other 3.